Okay, today we're going to talk about protein synthesis, going from a gene on the DNA to a protein. The flow of genetic information. The information content of DNA is in the form of specific sequences of nucleotides. The DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. And proteins are the links between the genotype and the phenotype. One of the last topics that we'll explore involves gene expression, which is a process by which DNA directs protein synthesis, including transcription and translation. Now, this image that's in your textbook is put there just to ask a simple question. How does a single faulty gene result in the dramatic appearance of an albino deer? Think about that. Now, how this occurs is that genes specify proteins via processes known as transcription and translation. There are many experiments in the past that fundamentally describe the relationship between genes and proteins. One of the most famous involves an experiment done on metabolic defects. This provided evidence that genes do indeed specify proteins. In 1902, the British physician Archibald Garrod was the first to suggest that genes dictate phenotype through enzymes that catalyze specific chemical reactions in the cell. He speculated that a disease known as alkaptonuria was a rare inherited genetic disorder of phenylalanine and tyrosine. The problems in metabolism of those. It's a hereditary disease caused by the absence of an enzyme that breaks down a specific substrate, in this case, alkaptonuria. Garrod suggested that the symptoms of such an inherited disease reflected a person's inability to synthesize a particular enzyme, and referred to such diseases as inborn errors of metabolism. Many research and experiments done later supported Garrod's hypothesis. Progress in linking genes and enzymes rested on a growing understanding that cells synthesize and degrade most organic molecules in a series of steps, a metabolic pathway. Now, in the 1930s, George Beadle and Boris Fressy speculated that each mutation affecting eye color in Drosophila blocks pigment synthesis at the specific step, a very specific step, by preventing the production of an enzyme that catalyzes that step. Now, neither the chemical reactions or the enzymes that catalyze them were known at this time. But Beadle and Tatum were able to establish a link between genes and enzymes by exploring the metabolism of a bread mold known as Neurospora crassa. Beadle and Tatum bombarded this bread mold with X-rays and screened the survivors for mutants that differed in their nutritional needs from wild-type mold. From this, they eventually developed a one gene, one enzyme hypothesis, which states that each gene dictates production of a specific enzyme. If you look at this closely, the question that we ask is, do individual genes specify the enzymes that function in a biochemical pathway? The story about what gene expression produces is an interesting one. They realized that some proteins aren't enzymes, so researchers later revised the hypothesis, one gene, one protein. Many proteins are composed of several polypeptides, each of which has its own gene. Therefore, Beadle and Tatum's hypothesis is now restated as one gene, one polypeptide hypothesis. Note that it is common to refer to gene products as proteins rather than polypeptides.